So when we talk about gene regulation, this is really helping us understand how all of our cells, if we take the human, for example, right, all contain pretty much the same DNA. So why do some cells, right, look different and perform different things compared to other cells? And so all of that is basically directed by gene regulation. So we'll take a little look at that, kind of give you a taste of all of those processes that are happening there in the background for that. So just a reminder, on Friday, we will be doing a team-based learning activity on the central dogma. So kind of helping us pull all of this together. And then I may give you guys some questions, depends on gene regulation, but we'll definitely leave some time for review. So if there's anything that if you have any questions or anything over the content that we've covered here in Module 6, that you want me to go over on Friday, feel free to post it in the Ask It basket. So maybe I'll start a chain there. So that way, if there are specific topics you want me to go over, I make sure that we have the resources ready to do that. And then just quick overview, what to do this week. So you have your homework 17. KDA 6 will open on Friday. You have the Process of Science Assignment 1. So this is where you watch or attend a seminar or a research presentation and fill out the worksheet that's provided on our website, on our As You Learn site. I do have this set to be submitted by Friday, but I did want to check in with you all and see if extending that to Sunday would help you, just kind of balance your schedules. I know only about, I think 6 or 7 of you have turned this assignment in already, so there's a lot of you still working on it. So if you think it would be helpful to extend the due date to Sunday for you, just throw something up in the chat and we'll decide at the end of class what to do with that. And then you should be working on your final presentation as well. All right, so today's learning objectives, so I have it kind of split into 2 parts. So we're finishing up translation. And so we're going to finish the last part of that. And we're going to talk very briefly about some of the post-translational modifications and look at how those kind of help, again, to get the proteins in an active form. All right, so we ended off on Monday talking about this whole process of translocation, where, remember, our ribosome was kind of moving down that strand of messenger RNA so that the transfer RNAs that were docked in the E, P, and A slots, right, they all shifted over by 1, which would open up that A spot so a new codon could come in. So this is, again, part of the elongation process of making our primary strand of our peptide. And so, again, just to remind us that the translocation does accomplish 3 different things. So the transfer RNA that has lost its amino acid or already donated it moves to the exit site, and then the the transfer RNA that has the growing chain on it will move then to the P site, therefore opening up the A site so we can get that new charged transfer RNA in. And again, when I use the word charged, I'm just indicating that it has an amino acid attached to it. All right, it doesn't really have to do anything with like positive and negative charges. It just means that it has an amino acid attached. So I know that's a little bit strange language there, so just wanted to remind us. All right, so in this picture here, and let me see if I can grab my pen real quick. All right, so we have basically this diagram is showing us this process, right? So this was, don't do this again. So looking at the picture here, this is showing us what's happening in a bacterial cell, but this process would be very similar in a eukaryotic cell. The only difference is that this methionine here is might be slightly different, right? So this is a modified one for the bacteria. But it had aligned here with our start codon. We had the second transfer RNA come in that corresponded to this second codon here, and we formed a peptide bond. So just as a quick check, is this enzymatic reaction here being facilitated by a protein or by RNA? Who remembers? You can throw it in the chat, or you can just unmute yourself and shout it out. So is this being catalyzed by a protein or by RNA? So remember, this was kind of special. Anybody? All right, thanks, Kristen. All right, now we got a bunch. All right, so, oh, we're divided. Interesting. Okay, so we got a lot of responses. So it's almost 50-50. And we got some questions, so we're not super confident. So remember, this was being catalyzed by the actual ribosomal RNA that is present here in the ribosome. So this made it what we call a ribozyme because it's catalyzing that reaction. And this is what led scientists to believe in the RNA world hypothesis that RNA actually was first, and then DNA kind of developed later. So this is very unique to the ribosomes, that the RNA is actually what is helping with the catalysis here of forming these peptide bonds in the ribosome. All right, so don't forget that ribosomes are ribozymes. All right, and so therefore that means the RNA is responsible. So thank you all for participating and putting out your best guess there. So great job. All right, and now the translocation step, we see that everything has kind of shifted over here and allowed this A site now to be available for our next transfer RNA. All right, so if we kind of look, for those of you who like just a quick short list of what was happening, you basically have the arrival of that charged transfer RNA into the A sites, peptide bond forms, translocation occurs. So these three steps get repeated all the way to the end until we hit that stop codon. So these three steps are kind of just like repeat, 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 and that will help form then that really long peptide chain. So whatever the instructions were that we got from our gene, that's what we're going to be making. And so this is just a continuation of that other figure we were looking at. So you can see, this was our first transfer RNA here now in the E sites. We can see that we have the glutamate attached to the methionine, and now we have an arginine coming in. We have our enzymatic activity occurring and forming a new peptide bond. The thing I want to draw your attention to in this picture is right here. So if you notice, the covalent bond that attached the amino acid to the 3' end of our transfer RNA is no longer there. So you see this black line in the first picture, it's no longer there. So when we attach the amino acid, that this glutamate here that's on the P site, to the incoming amino acid, during that process, we're going to also, it's not just forming a peptide bond, it's also breaking the covalent bond that held it to the transfer RNA. So there's two reactions technically that are occurring in this location. And what that allows then is for the transfer RNAs then to move to the next docking port, right, within the ribosome, so that then they can leave, right? And we still have the growing chain attached to one of the transfer RNAs. So just wanted to point that out, that there are technically two reactions that are occurring in this process here at step 5. All right, so like I said, that process just continues over and over and over again until we basically get to that lovely stop codon. So there are 3 of those that we have on our codon chart, right? And you will have that available to you on your KDA this weekend, so you don't need to memorize anything. You can simply look it up if you need to. So what happens is when we come across that 3-letter stop codon that is in our messenger RNA strand, right? When that aligns with the A site, rather than a transfer RNA coming in and docking, we have this look-alike protein called a release factor. So it looks very similar to a transfer RNA, so it still fits in that location, but it doesn't really it doesn't have an amino acid attached, and it's not made up of RNA. It's simply a look-alike protein that kind of docks into that A portion. And what it does is it will cause like a hydrolysis reaction to the bond that's linking the P site transfer RNA to that polypeptide chain. So if we go back, this connection that's right here. All right, so that, like I said, remember that bond typically gets broken when we add a new amino acid, but when we get to the end, I don't have another amino acid. So we're going to see that this release factor actually is going to help promote the breaking of that covalent bond so that we can release that polypeptide chain out into the cytoplasm, as well as release the transfer RNAs that are there, right? So the whole thing kind of just falls apart so that everything gets to be released into the cytoplasm. The peptide chain will continue on and doing its kind of post-translational modifications to get into its final functional form. And the other parts then can go and get reused and help to translate another messenger RNA strand. So again, if we just continue along that picture, we see here that we finally come to a stop codon. You can see here that we have this protein-based release factor that looks very similar to the transfer RNA, but again, doesn't contain RNA or an amino acid. And what it's going to do is help to hydrolyze that covalent bond that is attached between the amino acid here and this transfer RNA. So it has a specific function, right? It's to kind of break that bond. Once that bond is broken, like I said, all of this stuff can now be released into the cytoplasm, and the actual ribosome will come apart into its large and small subunits so that it can start that whole process over again. All right, is everybody feeling pretty good about this process? I know there's a lot of steps, and so it might seem a little bit overwhelming, I'm sure, but does it seem to make sense as we're kind of talking through the steps? Everybody, all right, I see Claire shaking her head, and Tyler and Kristen. Good, I see the top of Max's head there. Let's see if I got any more thumbs up somewhere in the class. So Elijah, you doing pretty good? Just gonna pick on some random people here. Mason, is this all good for you? And again, you can just, yeah, awesome, thanks for the thumbs up. All right, how about Ike? Haven't talked to you in a while. How's this information setting with you?
Yeah, it's good. I like it.
Awesome. Yeah, very good. All right. All right, so I do have a video in here. Again, this PowerPoint is posted just as a PowerPoint in our As You Learn course. So if you want to watch this kind of all happen in motion, this is a pretty nice little cartoon. So you can take a little break, I guess, and watch a short video on it. So I am not going to spend a whole lot of time talking about post-translational modifications, other than to say that this does happen, right? And we can tie this back to our chapter on proteins, right? So when we first learned about proteins and how they go from that primary structure to the secondary, tertiary, and quaternary structures. So all of that is part of this post-translational modification. All right, so that is an extensive series, right, of steps and folding and bending that take place. And one thing that we can add to our knowledge on that already, is that not all of these proteins are going to fold and get into their functional form at the same speed or at the same rate. And so sometimes that has a bit to do too, as we're going to see, with that gene regulation, right? So this is a spot where it can get held up a little bit if it takes a particular protein a long time to go through this process, right? So some of them may need to even add extra things, right? Like sugars or lipids or those phosphate groups that can activate and deactivate enzymes. So there's a lot of stuff that can take place here. So I just wanted to kind of mention that, but we're not going to like go back and talk about all of the different phases that the amino acid or the polypeptide chain goes through to get to that final functional form, right? So we did talk about that earlier in the semester. So if you do have any questions as you're reviewing that material, feel free to let me know. All right, so here is our summary slide, right? So we have talked about all of this stuff. So we started off with our gene in the nucleus. That's, just a part of that DNA. We made a pre-messenger RNA. We formed through the process of transcription. We then processed it to remove these introns and to put a 5' cap, then a 3' tail on it. And then once it was ready, moved into the cytoplasm and then found itself a nice little ribosome and was able to, go through everything we just talked about Monday and today. So again, this might be a nice picture for you all to refer to as you're kind of walking through the different steps of this process. So it can kind of help your brain associate a picture to a, an actual step. I think that helps a lot when there's a complicated process like this to have a visual to attach it to. All right, so you guys ready to talk about gene expression? This was, I think, is a very exciting aspect of, this whole process, but it gets so detailed. So if you are interested in learning more, it is in chapter 19. Some of the information that I have pulled, I did pull from a couple different sources, just so I could give you a more complete picture of the importance of controlling gene expression and where and why it's used, which our book didn't go into a whole lot of detail on. So what we're going to start off with is looking at situations. Oh, is it not showing full screen for you all? Let's see, is anybody else having trouble with this presentation not showing full screen?
It's just showing up in like your presenter mode with like all the slides at the bottom and.
Oh, that's awful. Okay, that is not what we wanted to have happen. Here we go. I am so sorry about that, folks. Is that better?
Yeah, that's better.
I clicked on the wrong, there's too many options now. I have a different setup, so I apologize. All right. So what we're going to look at is looking at different situations where gene expression is or can be regulated in this whole process that we've been talking about. So we're going to start off with that. And then we're going to look at points, where they're regulated. And then I'm going to throw at you a little bit of some new vocabulary, just talking about the difference between different types of genes, ones that are referred to as being inducible, ones that are repressible, and then ones that are considered constitutive, meaning that they're pretty much around all the time. So that's really what we're going to focus on here. There we go. So like I mentioned, regulation of gene expression is pretty complex, especially when we look at eukaryotes, right? So when we were talking about transcription with our prokaryotic cells last week, how we had the promoter and we had the sigma factor that kind of would help pull the RNA polymerase and the promoter region of our DNA kind of all together. So it's relatively simple compared to what we saw in the eukaryotic cells. And so I will bring back the picture of all the different places where we looked on a DNA strand to find all of these proteins that are going to help with controlling this expression. And so one of the reasons why, though, there's such a difference between prokaryotic cells and multicellular eukaryotic organisms is that if you think about it, we have a lot of different demands for our cells. So if you think about, you know, a single-celled organism, right, like a bacterial cell or an amoeba or something like that, right? It's just one cell, right? And that cell is going to be that cell. But if you look at us or a plant or an animal or any other multicellular organism, we're made up of thousands of different types of cells, right? And so how do we create all of these different types of cells, right? How does, you know, your skin cell know to produce keratin while your nerve cell knows how to produce neurotransmitters, right? Both of those cells contain all of the code to make all of those things, but yet they're only going to do what they're supposed to. So this is a really fascinating field, I think, to in biology is, you know, looking at developmental biology, right? And figuring out like, you know, what's going to turn this on and turn that off. So super, super exciting area. And this is why, you know, I felt it was important to talk to you all about this. So some of the reasons I tried to outline here that are important for this, what we call differential gene expression. And what I mean by that term is that we're going to, you know, only express certain genes in certain cells. That's what differential means. We're differentiating between the cell types and the genes expressed. So sometimes it helps them form into their specialized cells like we just talked about. Sometimes it will help those cells actually get together and form a tissue and maybe even later on an organ. It helps them to coordinate activity. So in a multicellular organism, right, we can't look at things in isolation. We have to know what's happening, you know, between one cell type and another. Your nervous system and your endocrine system are big examples of how they coordinate activity to other parts of our bodies to help with growth and movement, digestion, all of that fun stuff. Right? So you can really think of this as like a multicellular society, right? Living inside of you. So if you think about our, you know, society where we live, excuse me, we have, you know, people who do different jobs, right? So you have the, you know, people who run the grocery stores, and you have people who run the hospitals and the health clinics, and people who, you know, pick up the trash and run the electric plants, right? So we all have specialized functions, right? And our contributions to our society. And so we can kind of look at the cells inside of our body in a very similar way. All right. Just because I'm a picture person, I thought it would be kind of fun to kind of look at this. So we're regulating gene expression. So this is allowing all of these genetically identical cells here, right? So if I were to look at the DNA in these cells, it would be pretty much the same. The exception here is a mature red blood cell actually doesn't have a nucleus, and so it doesn't have DNA. But up until it becomes a fully mature red blood cell, it's going to share the same DNA as the rest of these cells. The sex cells over here are another exception. Right, these are not going to necessarily be genetically identical, right? You're learning in lab this week about meiosis, and that's helping us to create, you know, unique genetic footprints in these cells. But they all start from this common source known as a stem cell. So I'm sure most of you have heard of stem cells, right? Does anybody want to give a little, you know, 2-second definition of what a stem cell is, or what you know of stem cells? Because there are different types. Does anyone know what this kind of stem cell might be associated with? Or is this something that I'm like, oh, I don't know what you're talking about? And that's okay too.
I know that stem cells are kind of cells produced by the body that haven't been assigned for a specific function. So therefore they don't have the specific code of DNA for skin cells or neurons.
Yeah, you're definitely on the right track there, Tyler. Great job. So these would be, so if you hear like in the news, people get like a little bit upset, right? If you talk about embryonic stem cell research and things like that. So these are stem cells, and that's kind of what this cell here is actually representing. So if you start off with, a sperm meeting an egg, right? And it starts producing those very early cells. Those are our embryonic stem cells. And those cells have not differentiated into anything yet. Like they can become pretty much any cell in the body. But as they keep replicating, there's different factors that they get exposed to and produced. It's a complicated process, right? And some of them will start to form part of the nervous system or part of our digestive system or the muscle system, right? And so they start differentiating. So they start maturing into the cells that they're going to become. So just like we go through different life stage developments, I always like to think, when you're babies, your parents are like, oh, you can be anything you want to be when you grow up. And we don't know what you're going to become, right? And then as you start, growing up and learning new skills and finding out things that you like and don't like and are good at, right? You start off on a direction where you become this mature adult, right? And the same thing happens with our cells, right? So they start off as these immature stem cells that, the world is their oyster and they can become anything they want. And this is where this whole thing of gene regulation comes into play. So we're going to see that as they start down these different pathways, different genes are going to be turned on and turned off so that they can become that specialized cell. Let's see there. I'm just reading the chat real quickly. They're currently being used to regrow organs. Yeah, so there's lots of stuff. Yeah, that's a great example, Christoph. Yeah, so different stem cells. There's a lot you can do with it, right? So there's a lot of ethics behind using stem cells. We still have types of stem cells in us as we grow, but they've already started down a specific pathway. So sometimes you can take those cells, and if someone needed like bone cells, we could take those precursor cells and kind of try to generate bone cells. So that's kind of a fun area of research to look at too. Sorry, digressed a little. With that gene expression that we're looking at here, I just gave you like a couple little examples. Sorry, that was supposed to say a neuron. So there are a couple housekeeping, right? So there are housekeeping genes that pretty much all of the cells are probably going to be making, right? And so one of them, is probably the enzyme for glycolysis, right? So we want to be able to break down sugar molecules and produce some ATP, right? So we're going to see that pretty much all of our cells at some level are going to express enzymes that are involved in that process, right? So if you go back and look at our process of cellular respiration, a lot of those enzymes are going to be made all of the time. Now, if we look at things like neurotransmitters, right, these are only going to be produced in our nervous cells, our neurons, right? They're not going to be made by liver cells or muscle cells. So even though the muscle cells and the liver cells have that DNA in them to make the neurotransmitters, those genes aren't going to be turned on in those cells. So they're not going to go through that whole process of transcription and translation to make them. All right. And so this is what helps to give us this huge variety of cells that have very specialized functions. And where this regulation occurs within any given cell for any given gene can vary. So again, this is the picture we just looked at a couple slides ago, but I tried to put in a couple different areas here. So like if we talk about pre-transcriptional, this is actually looking at how the DNA has wrapped around those histone proteins when it condenses to fit inside of the nucleus. So depending on how that DNA has bent and folded over those histone proteins, right, it's going to give parts of that DNA more or less accessibility, right, to being transcribed. So that's one thing. So that's even before we can transcribe anything. How fast the rate of transcription actually occurs can also impact whether or not or how often a gene is expressed. Post-transcriptional, so how fast the cell goes and cuts out introns and adds 5' and 3' processing measures will impact it. How fast it actually goes through the process of translation. So that's not always consistent in how fast a cell may do that, or different cells do that. And then finally, we talked about the way in which protein folds and, some of that stability, which is dependent upon the protein folding into its final shape. So if we look at this really long list of things and we consider, cells try not to be very wasteful, which phase of this do you think is most regulated in the cell as far as, turning genes on or off? At which step of this process do you think this is happening at more frequently than the others? Does anyone have a guess to that?
I bet the pre-transcriptional or the post-transcriptional.
Yeah, great guess, Michaela. Yeah, so definitely this pre-transcriptional area here is definitely going to be an area where we see it, and yeah, possibly in that post-transcriptional too. But yes, this is going to be an area where we're going to see a lot of control over the gene expression. So we talked about that it can happen at any of these steps. What I'm going to try and do is briefly go over a little bit of chromatin, or just mention here that there's some chromatin remodeling that takes place. Our textbook goes into some very deep details on this. So if this is something that really interests you, feel free to dig deeper there. But this has to do with how the DNA in its chromatin form is wrapping around histones more or less tightly and exposing different types of side groups and things to make it more or less accessible to be transcribed. So there's a lot of stuff that can go on there. The RNA processing bit, again, that's what Michaela was just mentioning, really happens with that post-transcriptional part. And then the messenger RNA stability as it's leaving the nucleus, how stable have we made that so that it doesn't get degraded right away. So I mentioned, that transcription, really tries to conserve energy, because every step after the level of transcription is going to require a pretty big input of energy. And so if the cell doesn't need a specific protein, it's not going to waste materials, resources, and energy to make that protein, right? Our cells tend not to be wasteful. And so some of the terminology that you'll hear is that when a protein is not needed, it's shut off. All right. It just basically means we're not going to transcribe it. We're not going to make it. And so here are those new vocabulary words. These are not currently on a learning guide, so I just wanted to point that out to you all here. These inducible genes are ones that can be activated, right? So if a cell decides, oh, I do need to make X protein, there are certain other compounds available in the cell that will activate it. And so if you think about it, we talked about a situation in our cell where this happens. So if you think back to the cell cycle, can anybody remember, and you don't have to name a specific like activator type of molecule, but can you think of the process in the cell cycle where we would induce genes? We actually talked about a couple different ones. Does anybody remember anything as an example for this one? If I throw out CDKs and cyclins, does that jog memories? So our cyclins always present in our cell? So remember, they're a protein. Are they always present in a cell?
No.
No, they're not, right? Because that's why they're named a cyclin, right? Because they're only present. And so remember, and again, you don't have to remember all of the steps, but there's a protein that activates them and tells, our DNA, hey, I need you to make cyclin 1 or cyclin, G1S or whatever it was, right? So cyclins are a great example of these inducible genes, right? I don't want to make this, or I do want to make this particular thing. Repressible genes are the opposite. So they're going to be genes that get turned off by other things in the cell or things of that nature. And so again, we could probably go back to our cell cycle and look at that and find components of that where it's like, hey, I don't want the cell cycle to move forward. So maybe I'm going to stop this gene that activates that process, right? And so there's lots of examples that we could probably find if we went back and just looked at the chapters that we've studied, where we see, oh yeah, I see now, the cell has to activate this to turn this on, or, oh, we want to turn this off. And so we can group them into inducible and repressible genes. So these are ones we can turn on and off. And if anyone has ever seen one of those DNA like array chips, I don't know if maybe if you have like an AP biology teacher or somebody who did that, that you can look up images, and I should have done that, where things are turned like red and green in this mega data set, and it's showing biologists which genes are currently being expressed or repressed within a certain sample. So those are kind of cool to look at. And again, big mega data sets. It's a lot to sort through, but it's a direct application of being able to look at this in our cells. And then finally, the constitutive genes. And so sometimes I think that word is a little bit more like not intuitive for some students, like inducible. Yeah, okay, I get that means start something. Repress. Okay, that means I want to stop it. Constitutive means it's around all of the time. All right, so we find this a lot with many different enzymes or things that are just going to be present. If you look at our skin cells, the protein keratin is probably going to be considered a constitutive gene. It's something that your cells want to continually make because you shed your skin all the time, right? And so I need to be able to fill them with keratin, or we're going to look like really gross zombie creatures, right? So we want to make sure that that's happening. So you can think of constitutive genes help you not look like a zombie. All right, it's helping you produce that keratin and look, keep your skin healthy there. So I wanted to show you here what I meant by some of this chromatin remodeling and being able to access the genes. So to me, this is something that's a little bit hard to conceptualize sometimes. So you have, this 3-foot strand of DNA that is somehow wrapped and compressed and shoved into our tiny little nuclei, right? So we have all of this DNA, and within there, there's genes that are like, stuck in between these little, curves and stuff. So how am I able to, expose that? So we're going to see that during chromatin remodeling, portions of the, oops, I went too far. Portions of the chromatin can actually decondense. It's not going to do like the whole thing, but the area where we're interested in, or the cell is interested in, can decondense a bit and expose more of the DNA, so that we're able to find the promoters and the enhancers and all of that fun stuff, right? So this gets a little bit more complex. There are different things that do attach. So let's go ahead and move on to the next slide here real quick. So again, the whole purpose of this is just so that we have the cell's machinery has access to the very specific spots of the DNA that it needs, right? So that RNA polymerase has to come in order for transcription to start. And so that's why we have to unravel it. So it's not gonna be the whole thing. Again, just the parts that we're interested in. And again, that's regulated. And so we're gonna see that We talked about RNA processing. So again, just trying to build all of this together for you to kind of close the loop. We talked about this being unique in eukaryotes, that it gives us, removes those introns, puts the protection on the ends of it so that we have that nice mature messenger RNA that we can work with. But in that process, with that alternative splicing that it can occur, so depending where, which promoter and enhancer has, attached to that strand of DNA, we can actually get, splice out different regions, right? So the introns and exons may not always be consistent within a specific gene, right? So that's that whole alternative splicing craziness that takes place, which allows us to make different proteins from kind of that same strip of DNA, but again, after it's been processed. So kind of crazy stuff. Certainly makes studying some of those genes complicated, because if you don't have the right promoter, you might actually get the wrong transcript in the lab. So you have to pay attention to all of that. We want to make sure that the messenger RNA is stable so that it doesn't get digested by other enzymes right away once it leaves the nucleus, but also we don't want it to stay around too long and overproduce whatever protein it has, has the instructions for. So each one is going to have kind of its own, lifespan, if you will, that helps direct that. So looking at tying together the regulation piece and initiating transcription, now we can come back and talk a little bit more about those promoters, right? So we mentioned them back with like P53 and our cell cycle. So these promoters, again, are pretty complex. And usually each gene we're going to see has several regulatory sequences. So it's not just one thing that's going to bind to it and say, okay, we're good to go. It's usually a concerted effort by lots of different things coming in and interacting. We talked about kind of like this core promoter, which is the term core here is being used to kind of represent something that's very common in many, many genes. So many of our genes that we need to transcribe will have this TATA box, which is located upstream from the gene as part of that promoter complex. All right, so it's called a core promoter because it is found in so many of the genes. So it's core to the process. There will be a specific protein called a TATA binding protein that looks for that TATA box and helps, will bind there. And that kind of triggers the rest of the steps that we're going to look at. So TATA in some ways is kind of like, okay, this is where we're starting from. And so here's that crazy picture we've looked at a couple times. So if we look, we have this short promoter here in blue. That's upstream from our gene. But then we have some of these other things like enhancers, right, that are going to recruit other initiating proteins to help this process along. So we have some regulatory sequences. So again, these are going to be found somewhere in this DNA sequence that are specific spots where different proteins can attach and help initiate this process. So one of the first sequences that was actually discovered when they were studying galactose metabolism in yeast, so the breakdown of this particular sugar, was that they did find this core promoter. So something like that TATA box. It would be a little different here in yeast because that is a prokaryotic cell, but they were able to find that. Oops. In eukaryotic cells, we see that there could be multiple genes that are involved in regulating a particular DNA sequence. So some of the genes that are body needs or the cells need to transcribe actually can't get activated until other genes are activated to produce proteins to turn them on. So it's like super security, if you want to think of it that way. You've got to go through multiple layers of security to access maybe turning on this very specific gene. So an example of that, if we think back to P53, P53 was considered a tumor suppressor gene as well as a transcription regulator, right? And the reason for that is because the production of P53 actually initiates the transcription of other genes that are the ones that actually interact directly with the cell cycle. So it actually kind of fits into this model very nicely. So you can kind of think as co-regulation as super security, and you've got to go through these multiple steps of activating certain genes in order to turn on like the one that's going to go and actually do whatever it is the cell needs. We also have promoter proximal elements. And again, these are the purple ones here that are close to the promoter. And again, this tends to be a unique sequence as opposed to being like a Tata box that's common. These tend to be unique. And so that way we can get the specific gene of interest, right? That we want. All right. A researcher named I cannot pronounce their name. Tongawa, I'm not sure. But rather than studying yeast cells, they studied human cells and how their gene expression was regulated. And this was way more complex than those who were studying yeast, because this is where they learned about the introns and the exons, but then they also found enhancers, right? That were buried within the intron itself. So lots of interesting components here. So other things that they found were that there could be regulatory sequences thousands of base pairs upstream from where the gene was, right? So it wasn't enough just to look at, the genes, maybe 100 base pairs upstream. They had to make sure, like, if they wanted to really replicate what was happening in the cell, they needed to look, at a much, much larger strand of DNA in order to find all of the parts that were responsible for this. And so some of those things are enhancers. And so the enhancer is going to help recruit more proteins and enzymes and things to make sure that this gets transcribed faster, right? So the more enhancers there are, the more we're probably going to make of more transcripts we're going to make of that gene. So again, just kind of reiterating that. So the enhancers can be really far away from the promoter. They have discovered many, many types of these, and that a lot of genes are going to have more than one enhancer involved, and that sometimes they combine to more than one type of regulatory protein. So they're not necessarily a one-for-one type of thing like we see with some other regulatory events. They'll work even if their orientation is flipped or if they move to a new location. So if you remember when we were talking about that pie chart and mutations, and I was showing you like the parts, how much of the DNA was actually used to like transcribe a gene, this would probably fall in that large part. I think it was like 45% of all of the nucleotides that are in our DNA that are can translocate, they can move around. And so these enhancers can move to different parts of our DNA, right? It's not stagnant, which I think is another thing that's just mind-blowing, right? The DNA can move pieces around in order to, get certain genes to be expressed or turned off. So lots of very interesting things here to look at for sure. Let's see, I've only got these 2 slides. I'll just hit the main highlights here. So there are things that help with activating transcription. There are ones that help with repressing gene expression known as silencers. And so those are going to, help to turn off a gene if we don't need it. Again, the names kind of suggest what they're going to do. And then just kind of pulling it all together. This is just a reiteration of where we started, that these transcription factors, it's the presence of those within the different cell types that allows for a muscle cell to be a muscle cell and a nerve cell to be a nerve cell. So transcription factors are not going to be found in all of the cells, the same ones. And so depending on which transcription factors are found in which cell will determine which genes are going to be expressed and which ones are not. So that's kind of like the big picture here is, it has to do with all these transcription factors, enhancers and suppressors and silencers and all that fun stuff going on inside of your cell that helps it determine, okay, I'm going to be bone or whatever it's going to be. All right. So lots of stuff to study there. So I'm going to go ahead and stop our share so I can see all of you and see if there are any questions lingering. I can certainly stick around, but I know some of you got to get to class. Sorry, we ran over a couple minutes. But if you have any questions on this, we will have time on Friday as well to go over some stuff again. So don't hesitate to throw the questions up in the Ask It basket. So if there's something from this week or even last week that you're like, oh, still confused, don't hesitate to ask. This is a lot of material. It's cool, but I know can be a little bit overwhelming. So, all right, I will see you all on Friday.
Thank you.
Yeah, if you have any questions, we'll be here.
Thank you.
Thank you.
Yep.
I have a question. So my team, we emailed our person that